Hello viewers, hope you all are doing great. As you all are aware of ongoing protein series on my channel Foodscito, these days I am talking about physico-chemical properties of protein. So in continuation to that, today I am going to discuss about molecular weight and homogeneity. So whenever we talk about molecular weight of proteins, it can be determined using a number of techniques, namely light scattering, osmotic pressure, sedimentation and electrophoresis. Amongst these, electrophoresis is the most commonly used technique. So in today's video, I'll be discussing about electrophoresis in detail. Now, before moving on directly to the techniques, let's talk about certain basic things which forms the basis of electrophoresis. First of all, whenever we talk about proteins, they carry a free amino and carboxyl group. So due to presence of these free amino and carboxyl group, proteins are amphoteric in nature. Now the question arises, what do we understand by amphoteric? In the presence of alkali, proteins can act as acids and vice versa. This is amphoteric. Now, as you can see over the screen, I have represented a pH scale, how the protein solution behaves over a range of pH. Let's discuss. On lower side of pH, proteins are positively charged, while at higher pH, proteins are negatively charged. And somewhere around intermediate pH, they have isoelectric point. Now, what is this isoelectric point? Here, the net charge on protein molecules is 0. That means amino and carboxyl groups neutralize each other, thus forming a Zwitter ion as represented over the screen, right? And this isoelectric point, Zwitter ion, these are very important concepts from examination point of view. Now let's talk about electrophoresis. What does the name suggest? Electro, that means electricity is involved, right? Phoresis means separation. So how do you define electrophoresis? Migration of charged molecules in an electric field at a defined pH is electrophoresis. That's why I have discussed how the protein solutions behave over a range of pH. Now, what will happen when you apply electric field? Ions suspended between the electrodes will tend to travel towards the electrodes bearing opposite charges. That is, negative ions will move towards positive electrode and positive ions will move towards negative one. There are certain factors which affect the rate of migration of protein at a constant electric field. Now, what are they? Charge of the molecule, size of the molecule, and to some extent shape of the molecule. There are number of types of electrophoresis, but in today's session I will be discussing about two prominent techniques: paper electrophoresis and gel electrophoresis. Paper electrophoresis. This technique is widely used to demonstrate the presence of numerous proteins in small samples of biological materials. And it also gives an evidence of homogeneity or heterogeneity of a protein solution. So this paper is wet with the buffer solution in such a manner that one end of the vessel is in buffer solution with the cathode and the other end is with anode. So the protein is placed near the end of the strip. Now when you apply electric current, so under the influence of voltage, proteins move on the paper, and if it's homogeneous, proteins will move at the same rate. So they won't be separated into number of zones. And if the solution is heterogeneous in nature, proteins will migrate at different rates and thus will separate in different zones. And when this paper is suitably developed with the reagents that give color with proteins, the number of zones show number of proteins present in the solution. So this is all about paper electrophoresis. The other technique is gel electrophoresis. As the name suggests, electrophoresis, the principle will remain same: migration of charged molecules under the application of electric field, right, at a defined pH. But gel electrophoresis, that means a gel is involved. So most commonly used is agarose gel. Gel here will act as a molecular sieve. What does that mean? That means it, depending on the size, it will allow certain particles to pass through, while retaining others. So once you apply electric field, what will happen? Proteins will start migrating. So after the migration, certain bands will appear on the gel matrix at different levels. Those which lag behind indicate that they are heavier molecules, while those which move faster, they are smaller molecules. So that's all about electrophoresis. Stay tuned with Foodscito for more such concepts and learning in a simplified manner. अगर आपको ये वीडियो पसंद आया है तो प्लीज लाइक, शेयर एंड सब्सक्राइब. Thank you for listening.